name is Matthias Schamayer, and I'm very pleased to present you a recent study that was published in Gut. We were working in more than 20 years ago on inflammatory bowel disease and got interested in the susceptibility of infection by foodborne bacteria like E. coli. Enterohemorrhagic Escherichia coli and enteropathogenic E. coli are significant causes of morbidity and mortality, especially among neonates and the elderly. So this was the study led by Nadine Waldschmitt, who looked in detail how the mucosa was able to defend against this pathogen, and we used Citrobacter rodentium as a model to study the infection by this foodborne pathogen. This led to the identification of specific pathway required for defense against these bacteria. The specific defect for this signaling pathway led to an increased pathology in response to these bacteria, and we looked further in detail at what could be the mechanism underlying this phenotype. And she basically excluded the possibility that some bacteria from the gut microbiota may compete with the pathogen. And she also excluded the possibility that the changes in the gut microbiome resulting from the deficiency may also explain the susceptibility of these mice to the infection. To get further insights how this signaling pathway may regulate the response to the infection, she performed a molecular analysis and found a specific target that was basically not expressed in these mice. To better understand how this new target may explain the susceptibility of these mice to the infection, she performed a series of experiments. And first, she basically looked at the antibacterial activity of this molecule against the infection, and basically she was surprised to see that there is no effect of these molecules against the infection. She then looked in further detail how the immune response to the infection may be impaired in response to Citrobacter rodentium, and identified a defect in the IL-17 response that leads to a decreased recruitment of neutrophils that are important for killing this pathogen. In conclusion, these results unexpectedly found a new mechanisms involved in the response to this infection that is in charge of recruiting neutrophils at the site of the infection for clearance of this pathogen. And this led me to thank all the team members that were involved in this research program, as well as all the collaborators. Thank you.